So, how are you? So, after so long time, I'm again happy to share some views and recent, advancement of my science. What I am doing. Hope guys you are doing good. So let's talk about the recent interview where I have discussed about hypoxia, and this is very new dimension for my research. This is not my forte, so if I did do any mistake, please forgive me. But let's talk about this topic, which this really so much interesting to me specifically. That's why I'm discussing here. So let's talk about the hypoxia. So whatever the project whatever the interview that's have the project aim was this, and aim of the project is this, and this is the role of the project. So let's talk about the aim of the project. So understand how internal oxygen landscape arise in the root and how these spatial difference are sensed and integrated with the developmental programs. That work will focus on the mechanism operating and the interface to root tissue organization. So root development is very much important. So signaling pathways and environmental constraints using Arabidopsis as a primary model system. So basically I work on Arabidopsis thaliana. So that's why this project basically attracted me. So basically what the role of that interview जो भी वहाँ पे जाएगा उसको क्या रोल है, वो बेसिकली ये है कि how internal root environment influence developmental decision and tissue differentiation in the root, with particular emphasis on how signaling pathway respond to sustained change in oxygen availability. The role will contribute to defining how physical and biological features of root interact to modulate internal condition and coordinated growth. So it's a trade-off. Suppose it is a hypoxic condition. This is root and this is shoot. So root-shoot trade-off will take place during the hypoxia. So let's talk about this project. So first and foremost, I will congratulate that person, that Binay sir, for such a nice project that will be funded by the ERC. So how root breathe, that we have to understand. Okay, so just for understanding, just think, in the normal condition ethylene will be the normal, plant will happily live. Suppose plant is submerged or in the flooded condition, so ethylene will produce more and more, and it is the alarm for the plant that please look into that programmed cell death is going on by forming parenchymatous tissues into the plant system. So this is the ethylene, the hormone ethylene get trapped by the body and act as an alarm during the hypoxic condition, and through programmed cell death plant build parenchymatous tissue, and parenchymatous tissue is an alarm that yes, there is a hypoxic condition. So root oxygen dynamics and distribution into the root and shoot, as well as developmental signaling derived by oxygen reallocation reveal how O2 shape root development and architecture. This is I think the motto of that project. So I'm not going into that project details. So for this video I'm making for the general perspective to understand the hypoxic condition and normal condition, how plant behave in both the conditions. So what is hypoxia in a general perception? So it's a condition where root have inadequate oxygen due to the waterlogged condition or due to the light condition. I will explain, even to the higher altitude condition, whatever the, so oxygen is inadequate first and foremost. And what in major cases what happen, waterlogged soil is a major major case of hypoxia and leading to plant suffocation. So this is basically how we can understand hypoxia. Okay. So I will explain you a very very important pathway that is called endodermal pathway. Please go and read this pathway because this is very important to understand normal condition and hypoxic condition. In normal condition, this is ERF7. ERF7 is a ethylene responsive factor 7, class 7 transcription factor, which basically is inactive and it goes into the proteodermal degradation via the PCOS. This is oxidase enzyme which involve oxygen, and ultimately ubiquitination of this ERF7 factor takes place. That is the proteodermal degradation. But this is the normal condition. What happens in the hypoxic condition? In hypoxic condition, you can see here that this ARF 7 is get stabilized. How this is stabilized, I will explain in a very detailed mechanistic way. So this ARF7 basically stabilizes, and then what happens? It binds on this is this one. What is hypoxia responsive gene factors and elements? So if it bind, then hypoxia responsive gene will come into the picture, and hypoxia will prolong. So one of the paper that is published in PNAS by this group only, Binay Shukla et al., that stating that Suberin plasticity to developmental and exogenous cues to regulated by a set of 4 MIB transcription factor. 4 MIB transcription factor working in a particular in a proper fashion. So let's talk about this in a details mechanistic behind how this is happening in the plant system. So first and foremost, how the plant get transported into the xylem. For that you have to understand, this is 11th class question. Understand this apoplastic and symplastic way of water transport. In apoplastic water transport, it's going through cell wall to cell wall, and ultimately it will go into the pericycle, and then it loaded into the xylem. So what is the barrier? Endodermis is the barrier. Let's talk in a different condition. Suppose, what is symplastic? Symplastic means you have to spend money, ATP. So it's going through epidermis, cortex, endodermis, pericycle, and so forth. So this is with one cell to second cell to third cell. This is through the cytoplasm. It means there is involvement of mitochondria. So mitochondria will spend money, the ATP, and then the water transport will take place. Why this come into the picture, I will explain in a detailed mechanistic way. So why I am telling you this? This is very important to understand, guys. Just focus on that. That water and mineral basically absorbed from the root. Root will take this water and nutrient molecule to the epidermal cell, and then epidermal cell to cell wall to cell wall to cell wall to cell wall. Just see, this is the cell wall. One cell wall to the second cell wall. Here, and here, and it's going to. So barrier is this Suberin molecule, which is deposited in Casparian strips. So here apoplastic means of water transport is completely sealed. For that escaping this whole route, plant have evolved a secondary way to absorb this water transport. So in this transport mechanism, what will happen? Just think wisely. It will goes through one cell cytoplasm to the second cell cytoplasm via the interconnection that is the plasma destimata, and then finally it comes into the xylem vessel. So this is very interesting story. One, without any energy source, only through the diffusion process, cell wall to cell wall, cell wall to cell wall connection goes in, water transport takes place, mineral transport. But In the symplastic way, you have to spend money in the form of ATP. So I think here I make some clear indication that how this apoplastic and symplastic way of water transport takes place. So I have already explained those things, I will not discuss, so please go and check. So what is Suberin molecule? So Suberin basically a waxy substance biopolymers deposited into the endodermal cell. So 11th क्लास में या नीट में कई बार पूछा भी गया है कि ये बताओ जो कि मतलब कि कैस्पेरियस टाइप में कहाँ देखे जाते हैं, तो इंडोडर्मिस के पास पाए जाते हैं, और इंडोडर्मिस में जो मॉलिक्यूल डिपॉजिट होता है उसे हम कहते हैं सुगर. सो दिस इज वेरी इंपॉर्टेंट क्वेश्चन फॉर मेडिकल स्टूडेंट्स वेयर नीट स्टूडेंट्स वेयर दे आर ट्राइंग फॉर दैट. Okay, so I'm not going to that part, I will just discuss hypoxia condition. So this is very important to understand that paper. Okay, so Suberin molecule is okay. So blocking the free diffusion in the apoplastic and transcellular pathway, that is complete blocked. So this barrier forms after earlier lignin-based Casparian stripe which create a apoplastic seal. So Suberin deposition is highly plastic in nature because it's controlled by several factors. So it's developmental cues via the SGN3 CIF pathway. It's a very interesting pathway. Go and read that pathway, which basically monitor the CS integrity. Okay, the exogenous environment cues like nutrient, stress, hormone, ABA and abscisic acid. Also environmental factors like salt, ethylene. I have already talked that the hypoxic condition ethylene is higher. Okay, so this all thing include and induce the Suberin deposition. So this is all about the understanding of the paper. So I will try to make it crisp. So what is the take home message of this paper? Why I have kept this a NAM Q cook protein? Because this basically involving the lignin biosynthesis. Lignin is again in the very important for the vascular bundle and Casparian strips. So even these factors is on our promoter SQS1 where I have worked. You can check my paper. It's recently come to the plant physiology. So that paper talk about how this light regulating factors regulate squalene biosynthesis. That in detail biotic stress I am doing currently, but abiotic stress part was published in plant physiology. So go and read that paper, very nice paper. So author demonstrate that Suberin regulation occur independently via two pathways, that the SGN and ABA biosynthetic pathways shown through the genetic and pharmacological experiment. Okay, so this pathway, just I am going to summarize that they have used 4 MIB transcription factors, MIB 41, 53, 92 and 93. So if they mutated single mutant, they have generated here they have generated single mutant, then no phenotype was observed, and there is the Suberin deposition and the loss of Suberin molecule. But they have generated quadruple mutant of all these 4 mutant transcription factor. They come up with 78% reduction in this the polymer. So this mutant form Casparian stripe, how these are happening, they have given the very nice fine thing. That's how this transcription factor is very important for the regulation of various molecules in the plants. So this work provides a first clear transcriptional mechanism how these two independent signaling pathway of Suberin plasticity. The quadruple mutant is a major source, a strong specific Suberin deficient line without Casparian type issue. और इन म्यूटेंट में भैया एपोप्लास्टिक में से भी प्लांट को दो पानी चलेगा, कोई ज़रूरत नहीं है. सुबेरिन ही नहीं है तो फिर कोई बैरियर ही तो नहीं है. सुबेरिन ही तो मेन बैरियर है एंडोडर्मिस में. समझ में आया? सो दिस इज द पेपर that is published in PNAS. So for beginners, I will explain a little more about this constraint. Okay, so let's talk about the hormone regulatory coordination with the developmental program stress responses. So this is the hypoxic condition for the root. So first I will talk about this part, that is the leaf system. This is the stomata. So stomata opening and closing is being regulated by ABA hormone, that is abscisic acid. So in hypoxic condition, stomata got closed. Why? Because ABA is not acting in a proper way. But in recovery, this is opening. See A. Okay. So this is opening. So in recovery, ROS generation is very high, which triggers abscisic acid to open stomata. So this is the A story. Okay. So ABA signaling cascade hypoxia and recovery condition. The core abscisic acid signaling pathway is activated in hypoxia during the post hypoxic recovery during the induction of stress responsive gene and restoration of stomatal function. So second one is a phytohormones, IIA, ABA, ETG. What is this? Oxygen, abscisic acid, ethylene, gibberellic acid. All these hormones is coordinated during the hypoxic condition and help in shoot elongation during the submergence condition. Okay. So phytohormone regulates shoot development under the hypoxic condition. Hypoxia reprogramming shoot architecture by modulating stem elongation, shoot branching, lateral bud, outgrowth, dynamics, hormonal crosstalks. This fine-tuned interplay ensure a adaptive shoot morphology under the hypoxia condition. Okay. So now C. So C को बड़े अच्छे से समझने की कोशिश करो. So hypoxia induce root remodeling and metabolic hormonal integration and also induce expression of ARF7 related to AP2. So it's a relate ARF7 कहीं पे देखोगे कि ये भी RAP2.12 is related to ARF7. So this as equivalent, not the same, but this is equivalent. इसके और भी homolog हैं, which limits lateral root formation. Okay. So think wisely here. Okay. So in this case, what is happening? Just see. This ARF7 that is equivalent to this, so which inhibit lateral growth development during the hypoxic condition. ROS is very high, adventitious root, parenchyma formation because the programmed cell death is going on there. So this is happening. Now the thing is, what is happening to these lateral root formations? Ethylene is higher, so ethylene will be higher, then it will increase the level of pin 3, pin 7 and lateral root formation will restricted. Apart from that, when ABA come into the picture, it will increase the lateral root formation via the MIP transcription factor. ARF come into the pictures. See, this is very finely tuned. So these all hormone have a great importance during the hypoxia, how they manage. So it's very important and contradictory part. And one thing which always a key factor to attract me, that is the secondary metabolites and metabolites, primary and secondary metabolites, homeostasis. So I will I will dig more in this perspective. Think what is, if hypoxia is there, SNRK is a sugar kinase, TOR is again a very important factor that regulating the sugar kinase and sugar homeostasis in carbon source. This is a hexose kinase. So these all enzymes basically how they are regulating the hypoxic condition with the hormonal integration is still unexplored. This is the area. अगर हम कहें तो एक बहुत बड़ा एक प्रोजेक्ट बनाया जा सकता है इस एरिया से कि कैसे, अब सपोज कि समझो कि मैं हाइपोक्सिया में, फील इट, जस्ट क्लोज योर माउथ एंड ऑक्सीजन लेवल विल ड्रॉप. What will happen? You will suffocate suddenly. So in that case, if waterlogged condition, high altitude, light dark condition, so hypoxia is induced. Think wisely. During the hypoxia, carbon source sugars are not available. Sugar is not available for the plants. Then what is available for the plants? Plants have available these the molecules. Which molecules? TCA cycle intermediate, which I will explain in the subsequent slide. So hypoxia induced root remodeling and metabolic hormonal integration also induce expression of ERF related, which limit the lateral root formation. So several transcription factors further modulate the expression of gene involved in IIAET, ABA biosynthesis and signaling to the balance of lateral root development and root meristem maintenance. Furthermore, sugar sensing. अभी मैं बताया ना, sugar is very important. Sugar sensing molecule like TOR, target of ripamycin, SNRK, हमने पहले बताया sucrose, non-fermenting related protein kinases, HXK. So this is a hexokinase. It's again a kinase. Suggests a metabolic hormonal nexus in the root adaptation. Root have to adapt in the hypoxic condition. So, however, the mechanistic detail of the crosstalk remain largely unexplored, warranting a future investigation of how this sugar signaling fine-tuned during the hypoxia responsive growth. This is again very interesting area. So just I just want to make attention here that hypoxic rice germination. Suppose rice कब germinate होता है? पूरी जो फील्ड होती है फ्लड कर दी जाती है पानी से, अब वहाँ से इमरजेंस होती है, तो वो भी तो हाइपोक्सिक कंडीशन है. जो एम्ब्रियो रखा हुआ रहता है, तो जो हाइपोक्सिया के कारण ये सारा जो ग्लूकोज अब नहीं है, तो अब क्या यूज होगा? तो फॉर दैट यू हैव टू अंडरस्टैंड द दिस. Here everything will be clear. So darkness, starvation and ethylene, age dependent leaf senescence leading to the programmed cell death, chloroplast, autophagy to form erenchymatous tissue, erenchymatous cell, erenchyma cell to float on the surface. Plant is adapting in that environment of the hypoxia. So अब कार्बन सोर्स तो बचा नहीं. If carbon is not available, then what will happen to the plant? Then this will use this intermediate of the TCA cycle, like acetyl coenzyme is the first step for the fatty acid biosynthesis also. It also goes into the terpenoid biosynthesis and so on and so forth. MBA pathway is basically start with acetyl coenzyme. Sugar is not available because this is the fermentation case. There is not 36 ATP, there is only 2 ATP. It's a fermentation process. Hypoxia is not an aerobic process, this is anaerobic process. So think all over your 11th, 12th biology and whatever we can say the biomedical part. So it will be very interesting factors. We as a metabolites engineer, metabolic pathway engineers, we always think about what will happen when we divert the flux. यहाँ पे भी तो फ्लक्स डैमेज नहीं होगा. अब कार्बन सोर्स बचा नहीं, शुगर है नहीं. सर केल्विन साइकिल चल ही नहीं रहा भाई. यहाँ टीसीए साइकिल चल के ये सी इंटरमीडिएट को यूज करेगा बिकॉज वहाँ एरोबिक चीज़ें है ही नहीं. ओके. अब जो एक बड़ा इंटरेस्टिंग फैक्टर है, बहुत लोग कहते हैं ठीक है भाई हाइपोक्सिया हाइपोक्सिया हाइपोक्सिया, क्या प्लांट डेली लाइफ में हाइपोक्सिया फील करता है क्या? यस. In daily life, day and night shift have a drastic importance on the hypoxia condition and normoxia condition. In day, that is the normoxia condition, and at night, this is hypoxic condition. So it's very interesting because I work on light signaling, so I completely agree with that. So you can stop here. You can read that the down whatever the written here in the section, that what is this? Young emerging leaf experience oxygen fluctuation under day and night. क्योंकि जो young leaf है उसको समझ में नहीं आ रहा है हो क्या रहा है. अचानक सन आ गया, सब कुछ आ गया, लाइट है, सब कुछ ठीक है, अचानक वो गया. क्योंकि नॉर्मल डे में नॉर्मेक्सिया चल रहा है, क्योंकि ये ERF7 विल डिग्रेडेड, प्रोटेजोमल डिग्रेडेशन होगा. ठीक है. नाइट की कंडीशन में भैया वो है ही नहीं. अब ERF7 स्टेबलाइज हो गया, वो जाएगा न्यूक्लियस के अंदर और हाइपोक्सिया रिस्पॉन्सिव जीन को बढ़ाएगा. ठीक है. सो ERF7 आर स्टेबलाइज, ट्रांसलोकेटेड इनटू द न्यूक्लियस एंड ट्रिगर द ट्रांसलोकेशन ऑफ हाइपोक्सिया रिस्पॉन्सिव जीन. This ERF7 dependent cyclic mechanism create a metabolic switch and slow down the respiration rate, during the night. So it's again very important for me, specifically me, because I work on light signaling. So one paper that is in the scientific report, here, human nature is always, anthropogenic activities, always, danger for the ecosystem when it is not in a proper fashion. If it is not well regulated, so what will happen? So in this paper, author suggested a artificial light at night, ALAN, which is the short form. which क्यों ऐसा कह रहे हैं, सब थोड़ा समझो. हम लोग क्या कर रहे हैं ना, हम लोग विकसित होने के चक्कर में ना बर्बाद होने के कगार पे पहुंच गए. अब कोस्टल रीजन में गोवा एरिया में चले जाओ घूमने के लिए, तो रात में इतनी लाइटिंग होती है, इतनी लाइटिंग होती है कि आसपास के इन्वायरमेंट में दिन ही फील कर रहा है भाई. आर्टिफिशियल लाइट आप दे रहे हो. सो इट्स ए स्ट्रेस, लाइट स्ट्रेस. So light stress will completely change the plant circadian. So plant circadian will change, then what will happen? So this I will not read all this section, but one line I will जरूर मैं यहाँ पे बोलना चाहता हूँ कि marine organism in the lagging behind at that the terrestrial one that affect on marine primary producer are the most unexplored, where we are disturbing the whole ecosystem. What we are doing? We are disturbing the the producers, which is the major source of oxygen to this anthropogenic activity. So whole circadian mechanism will change. JTL family, JTL loop, regulatory factors. Think about the JTL family. What is the importance of plant circadian? So whole thing we are disturbing. So what will happen? Author shows that reduced growth in the seagrass leaves when these were transferred into the controlled condition and without lighting during the night. So this is very important paper. Please go and read that paper, how the anthropogenic activity is disturbing this cyclic hypoxia condition. We are generating the plant with, we are disturbing the plant the normal condition, because this is daily phase. Normexia and hypoxia is also important for the plant for the survival. What is the key take home message? Because the light and dark is very important for the plant. If you will disturb the circadian, everything will change. So I will not go in the philosophy class, because I am discussing about the phytohormone and how these are regulated. So this paper, it's a very fine-tuned paper that I am sharing all these reviews on the factors. So think, this is the normal condition. GA, ABA, IA, this gibberellic acid, abscisic acid है ना, above the ground IA ये सब है. below the ground क्या है? jasmonic acid है, ethylene है, salicylic acid है, oxygen है. These are working in normal fast. Nothing is bad. Everything is okay. But if hypoxia comes into the picture, अब देखो below the ground, नीचे रूट को देखो. अब क्या हो गया? इथिलीन बहुत ज़्यादा है. अब क्या करेगा वो? एरेनकाइम में टिश्यू बनाना शुरू कर. कैसे? यहाँ पे देखिए, ये जो है ये एक तरह का ऑक्सीडेज है. कैसा? बस्ट ऑक्सीडेज. पढ़िएगा इसके बारे में. ये ROS को बनाएगा. और मैंने बताया था कि रिकवरी फेज जब आप हाइपोक्सिया से जब लेके आएंगे प्लांट को, आगे भी दिखाऊंगा, पिक्टोरियली भी दिखाऊंगा कि कैसे जब आप हाइपोक्सिया से बाहर भी प्लांट को लाते हैं तो ROS कम नहीं होता, बढ़ता ही जाता है. बिकॉज हाइपोक्सिया का क्या है? इम्पैक्ट रिकवरी के बाद भी प्लांट सही से नहीं चल पाता है. सो एज ए रिजल्ट प्रोग्राम सेल डेथ होगा. इथिलीन बढ़ेगा तो प्रोग्राम सेल डेथ करेगा. फिर जैस्मोनिक एसिड लैटरल एंड एडवेंटिटियस रूट डेवलपमेंट में काम करेगा. वहाँ पे इंड्यूस लैटरल रूट ROS स्कैवेंजिंग. अगर IA वहाँ पे बचा रहेगा तो स्कैवेंजिंग करने की कोशिश करेगा, ROS को कंट्रोल करने की कोशिश करेगा ताकि कम से कम इथिलीन का इफेक्ट दिखे, क्योंकि IA तो वहाँ पे बहुत ड्रामेटिकली चेंज हो. फिर अगर हम एपिकल मेरिस्टम की बात करें, सूट एपिकल मेरिस्टम, रूट एपिकल मेरिस्टम, सूट एपिकल मेरिस्टम. तो सूट एपिकल मेरिस्टम में you have well known theory of clavata and Russell. This gene will get disturbed. Even stomatal closure opening is regulated by abscisic acid and ethylene. There is, I have already discussed. So this whole thing will change. So I will remove this, that you can read, you can stop here and you can take the picture of that snapshot so that you can understand what this basically means. So okay, so again what I have told you previously, I will explain this. What will happen in the hypoxic condition? सबसे पहले तो dissolved oxygen है, DO, dissolved oxygen will drop drastically. If you will reoxygenate, then dissolved oxygen will not that much prominent. Ethylene, what will happen? Ethylene will gradually increase during the hypoxia, but other hormone will come into the picture and it will ultimately, ethylene will get decreased due to the action of IIA into the root system. ABA and jasmonic acid जब आप डालेंगे, सैलिसिलिक एसिड तीनों ABA, J एंड सैलिसिलिक एसिड, कोई फर्क नहीं पड़ेगा टिल क्या हाइपोक्सिया में वो धीरे धीरे डिक्लाइन ही करेंगे. जब आप जैसे ही आप रीऑक्सीजनेट करेंगे, फिर से ऑक्सीजन will come into the picture, it will drastically increase. It's a defense response for the plant. हाइपोक्सिया से बाहर आया है, अपने आप को जिंदा रखने के लिए वो भी बहुत कठिन हो जाएगा, क्योंकि उसने पूरा कार्बन सोर्स ही चेंज कर दिया. अब फिर से उसको शुगर पे आना पड़ेगा, तो पूरे पार्टिकल पूरा शिफ्ट होगा. समझ रहे हो? अब ROS की कहानी और अलग है. ROS इनिशियली बढ़ेगा बढ़ेगा बढ़ेगा, फिर IA उसको स्कैवेंज करेगा तो थोड़ा कम हो, जब आप रीऑक्सीजनेट करेंगे तो और बढ़ जाएगा. RAP 2.12, what is this? This is ERF7 like transcription factor. So what is happening here? If you introduce hypoxia, it will increase. Will reoxygenate, then it will drop. So ADH1, PDC1 and HV1, these all are hypoxia responsive genes. So what will happen? Okay, so this is the clear picture of how plant responds to hypoxia and reoxygenation response. So this is the normexia condition, normal condition. This is the hypoxia condition. This is the plasma membrane lipid bilayer. So this is the lipid. This is nothing. PA, PE, these all are lipids. So hypoxia condition में मैंने पहले भी बताया कि कैल्शियम सिग्नलिंग विल बी हाई. सो कैल्शियम क्या हो जाएगा? बहुत हाई हो जाएगा. कैल्शियम हाई हो जाएगा तो इसकी स्टेबिलिटी और इंक्रीज करती जाएगी, हाइपोक्सिया और प्रोलॉन्ग होगा. थिंक द नॉर्मल कंडीशन. दिस ERF7 binds with ACBP1/2 normally. So this is in control. This is get disturbed in the hypoxic condition and drastically transported to the nucleus. In nucleus, it's bind with the MD2 to stabilize. More stabilized form is this, and then hypoxic response is again prominent. In normal condition, what happened? This the stabilized form is get destabilized, and from nucleus it's going to cytoplasm. So cytoplasm में फिर यहाँ से बाहर फेंक दिया गया. यहाँ देखो, यहाँ से फिर बाहर फेंका गया. अब इसको प्रोटियोजोमल मेडिएटेड डिग्रेडेशन PC हो जाएंगे, ऑक्सीडेज जाएंगे, है ना, और ये सब धीरे 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 लाकर इसको 26x प्रोटियोजोमल डिग्रेडेशन करा देंगे ERF7 का. So this is the normal condition. This is the hypoxic condition. Hope you understand very well the story. So I will tell you some interesting fact that this is the wild type having 21% oxygen, that is the normal oxygen, 1% oxygen in the hypoxic condition. So hypoxic condition, root length drastically decrease. Okay. So to make you more picture, broader aspect. So this is the J come into the picture. So it's also showing that if you induce hypoxic condition, the root length will decrease. But one very interesting fact is one is JAR1. जार वन इज ए गेट कीपर, आपका गार्ड है हाइपोक्सिया का। कैसे? ये देखो, जब जार वन के म्यूटेंट में इन्होंने हाइपोक्सिया जनरेट किया, इनको रूट इन्हिबिशन नहीं दिखा। रूट इन्हिबिशन क्यों नहीं दिखा? क्यों? क्योंकि जार वन गेट कीपर था, वो है ही नहीं भाई। चाहे हाइपोक्सिया दे लो चाहे नहीं दो, कोई मतलब नहीं है भाई, ठीक है। तो इनसेंसिटिव ग्रोथ है। जार वन म्यूटेंट, what they are showing, do not inhibit the root growth under low oxygen, suggesting that jar one is a gatekeeper. If you remove the gatekeeper, anybody will come to your house. So this is the scenario. Okay. So now I have taken some important snapshot of the paper for you guys to deepen your knowledge like me, because this is completely new thing for me, but I really thank Dr. Binay sir for giving me chance to study these things. So designed to breathe, synthetic biology application in plant hypoxia. So read that paper, you will understand how can we design to breathe. So this is the main title of Dr. Shukla. Okay. So in another paper, it's a very nice paper, you can read that paper. They have, this is a review. In this paper they have shown a lot of things. I have also taken some snapshot of during the explanation part. I have used that paper that published in PCE journal, I think. So how this hormone ethylene, oxygen under hypoxia, how they are enhancing ACC, because ACC or ACS है ना, ये क्या है? ये सब आपके इथिलीन बायोसिंथेटिक जीन है। मेथियोनिन के बाद ये सब आएंगे सारे। However, because of ACO require oxygen, ACC reproduce hypoxic route transported through the aerobic tissue for the conservation of ethylene. क्योंकि ethylene is very important for the plant. Hypoxia में तो बहुत ज़्यादा बनेगा, तो फिर प्लांट अगर कोई चीज़ बहुत ज़्यादा बनेगा तो उसका रिवर्स एक्शन में ट्रेड ऑफ है ना भाई। कितना कोई चीज़ बनेगा तो एवरीथिंग शुड बी इन ए फाइन बैलेंस्ड। प्लांट ऐसे नहीं कर सकता। हाँ, रीऑक्सीडेशन फर्दर अप रेगुलेट ईटी बायोसिंथेसिस में जीन आया, ये बायोसिंथेसिस बाय द ट्रिप्टोफान। पता है ना ऑक्सीजन बायोसिंथेसिस कैसे होता है? ट्रिप्टोफान कम्स इन जो पिक्चर, वहाँ पे यूका जीन आता है। यूका जीन कैसे बायोसिंथेसिस में करता है, आपको ऑक्सीजन बायोसिंथेसिस पढ़ना पड़ेगा, है ना। तो ट्रांसपोर्ट के न्यूट्रिएंट होते हैं, बायोसिंथेटिक के न्यूट्रिएंट, रिस्पॉन्स। तो हार्मोन ना बहुत अपने आप में एक बहुत बड़ा की प्लेयर है। बायोसिंथेटिक जीन के म्यूटेंट अलग तरह से बिहेव करेंगे, ट्रांसपोर्टर के म्यूटेंट अलग तरह से बिहेव करेंगे, जो रिस्पॉन्स जो देखेंगे, रिस्पॉन्स म्यूटेंट अलग तरीके से दिखेंगे, है ना। सो generalized diphosphate is again very important for carotenoid, terpenoids biosynthesis. So this is again is trade-off between primary and secondary metabolism here. ET signaling plays a central role in hypoxia, generating tolerance, stabilizing EIN3, EINL1, activate and promote the stress tolerance, where this all key player takes place. Additionally, salicylic acid can independently stimulate root adaptation under the waterlogging condition. Overall, OT fluctuation orchestrated tight, in interconnected context dependent hormonal network regulating the plant stress responses. So hope I have given the crystal clear idea. You can read, I will just for your better readability I will keep this here. Okay, so you can take this snapshot and you can read more about how this ethylene jasmonic acid trade-off and crosstalk will influence both through microRNA, through different transcription factors, and like jazz, repressure, and how they are coordinating. So one paper, tool to understand hypoxia response in plant. इसको हम एक tool भी के तरह भी यूज कर सकते हैं. In this paper you can understand how we can use sensors, biosensors, to understand the hypoxia condition. Another paper, surviving in the floods. हियर सबमर्जेंस में क्या होता है, सब वन जीन जब गया प्लांट में तो प्लांट की हाइट बढ़ गई। ऐसा क्यों हुआ कि इसमें जीए की बायोसिंथेसिस बढ़ गई। अब जीए क्या करेगा, सूट इलोंगेशन करेगा, प्लांट क्या करेगा, जहाँ पानी दिखेगा उससे और थोड़ा ऊपर जाएगा, थोड़ा और ऊपर जाएगा, थोड़ा और ऊपर जाएगा। देखो यहाँ पे देखो नॉर्मल प्लांट, बट सबमर्जेंस प्लांट देखो तो प्लांट हाइट कितनी बड़ी होगी। ओके, सो इथिलीन यहाँ पे फिर अलग अलग आएंगे, और इथिलीन आएगा, काइनेजेस आएंगे। DWF इज ए वेरी इंपॉर्टेंट जीन ऑफ व्हाट, स्टेरॉल बायोसिंथेसिस अगेन। सो ग्रोथ और इसमें ट्रेड ऑफ फिर शुरू हो गया। So one very important paper is hypoxia is not only to the waterlogged condition, it's also influenced by geography, altitude, agriculture practice, day and night practice. Hypoxia can be generated through various means of transport. Even the hypoxia, जब हम एक पेपर पढ़ रहे थे तो उसमें देखा कि they are working on tomato model. अब टोमेटो में अगर आपसे पूछूं कि हाइपोक्सिया अगर आप क्या होता है, ग्रीन होता है, ब्रेकर होता है, और रेड राइप टोमेटो है, ऐसे ही होगा ना। तो ग्रीन में क्या है, हाइपोक्सिया ज़्यादा कम होगा, जैसे जैसे ब्रेकर में हाइपोक्सिया और बढ़ेगा। ग्रीन में ऑक्सीजन का ट्रांसपोर्ट तो वैक्सी ले रहेगा तो उसमें और ऐसे कमेगा। और ये जो पेडिसिल देख रहे हो आप, ये है ऑक्सीजन का ट्रांसपोर्ट का मेजर सोर्स। वेरी नाइस पेपर इन प्लांट फिजियोलॉजी, यू कैन इफ यू, आपको अगर वो पेपर चाहिए तो आप हमें कमेंट कीजिए, हम आपको पेपर भेजेंगे। Go and read how this oxygen, even hypoxia in the fruit is well coordinated, how this hypoxia driven the fruit ripening process, governing the ripening process, fruit ripening. Okay, so please go try to read that paper. Okay, so this paper telling you the geography, altitude, agriculture and hypoxia. These all are the recent study. I am showing you in very nice reviews, which whatever published in plant physiology. So again, very important topic is advancing the seed hypoxia research. So go and read that paper also. So apart from that, some here I am showing the nature part, that they have published some very nice article that how plant monitor integrate their barrier by sensing gas diffusion, that is the oxygen. Calcium signaling in the hypoxic response. I have already explained you that how this calcium signaling triggers during the hypoxia. So this is ABA, how एक बहुत बढ़िया थ्योरी पहले बताई गई थी कि जो ये लॉन्ग डिस्टेंस सिग्नल है जो ABA का, इट्स हैपेंस फ्रॉम रूट टू द शूट, including stomatal closing during the hypoxia. But this is not as like that. Surprisingly, grafting studies were used in the abscisic acid mutants, revealed that root-derived ABA is not essential for the response. It means क्या? नहीं भाई, ऐसा नहीं होता है। रूट से शूट में जो ABA का जो ट्रांसपोर्ट है, आई थिंक नहीं होता है। आई थिंक हार्मोन पर्सन कैन कमेंट ऑन दैट, and if you will comment and if you will correct me, I will always thank you. So please, if somewhere, because this is a hardcore hormone signaling involved here, because I am a metabolic engineer, so I pardon me if I am doing anything mistake. Please comment behind on the video. Is mediated by multiple receptor, including this PYR1, PYL related protein, leading to the activation of SNRK kinases. हमने कहा था कि sugar metabolism, that है ना, transcription stress responsive gene involved in promoting the appropriate plant growth. Under the hypoxia, ABA fine-tune lateral growth by modulating cell division and expansion, balancing the development and with the survival. These dynamics may also influence early structure and barrier linking ABA-mediated root adaptation. So this finding, for the different, different fruit demonstrate that the complex and tissue-specific role of ABA is significantly different among the crop species. And in response to waterlog, notably the extensive research and considerable changes have been conducted to identify how this hormone regulator responds against the low oxygen. This conclusion offers insight into the hormone regulatory network. Still further research is required to fully understand hormonal control. हमने अभी भी बताया कि कैसे शुगर का मेटाबॉलिज्म इन हार्मोन से कोऑर्डिनेट होता है. शुगर तो है नहीं भाई, शुगर पिक्चर से गायब हो गया. इन वाटर लॉक कंडीशन में कैसे एक्ट कर रहा है. So this is again for a general perspective for the students, that plants die from water overwatering because it create low oxygen condition that suffocate them. सबसे बढ़िया मॉडल है टोमेटो. टोमेटो को ज़्यादा पानी देके देखो, मतलब आप उसको जिंदा नहीं रख पाओगे. तुरंत आपको सारे रिस्पॉन्स, बिल्टिंग वगैरह सब दिखेगी भाई. ड्राउट के सारे रिस्पॉन्स आपको वाटर ओवर वाटर से भी मिलेंगे. अगर नीचे नहीं देखे ऊपर देखे, ऊपर भी बिल्टिंग दिखाई देगी ओवर वाटर कंडीशन से. इसको इपिनेस्टिक रिस्पॉन्स बोलते हैं. अगर हम एक फैक्ट्री माने, रूट एनर्जी फैक्ट्री को देखें, सो बेसिकली क्या होगा. कार्बन सोर्स लीफ देगा फूड बना करके रूट को, और सॉइल से एपोप्लास्टिक से प्लास्टिक वे में क्या किया, ग्लूकोज सॉरी मिनरल्स और न्यूट्रिएंट वगैरह नीचे से लिया. ओके, सो absorb oxygen from the soil and glucose from the leaf. This is the first step. This is called the intake. दूसरा क्या है, अब सेल रेस्पायर करें. तो सेल रेस्पिरेशन यूज टू ब्रेक डाउन दिस द कार्बन सोर्स. अब कार्बन सोर्स जैसे ब्रेक डाउन होगा, रेस्पिरेशन होगा, इट विल अल्टीमेटली व्हाट विल हैपन, एटीपी विल प्रोड्यूस एंड एनर्जी विल. सो दिस एनर्जी इज अ करेंसी. नाउ जब पैसे हैं तो आप एक्शन कर सकते हैं. So ATP power activate uptake of water and nutrient more and more. So this factory works in that proper fashion. But in hypoxia, every machinery got hampered. अब ऑक्सीजन नहीं मिल रहा है सही से. सपोज इस चीज को समझो, ऑक्सीजन नहीं मिल रहा, what will happen? वो ऑक्सीजन लेगा नहीं. ऑक्सीजन जो है इनएडिक्वेट है. अब ग्लूकोज ब्रेक डाउन नहीं होगा, रेस्पिरेशन नहीं होगी, एटीपी नहीं बनेगा, एनर्जी सोर्स नहीं मिलेगा, पूरा चेन डिस्टर्ब हो जाएगा. So if the whole chain got disturbed from step 1 to step 4, action, how can this action be completed? So this is again very important term to think on all those processes. This is the survival or growth. Survival or growth. It always takes trade-off. So I will not go into the how this electron transport chain happens and how this regulates both, the stomata, lumens, and how they arrange ATP because this is very important for the, if you are preparing for PhD from India, then you need to think on all those perspective, because this is very important for the direct perspective. Okay, so as I have explained you that in the hypoxia condition, if during reoxygenation response, ROS generation will be always higher. So if ROS is higher, this lipid of peroxidation response is higher. So MDA will be higher. So you can measure this oxidative stress through measuring the MDA content and other things. So this is all about, I think, for today. Hope if I missed something, I'm really sorry for that, and hope you will understand whole scenario, and thank you so much for watching. See you soon into the next episode. Sorry.